Welcome friends. Before getting into the discussion of nuclear division, that is mitosis and meiosis, today we are going to talk about a very basic discussion on chromosome concentration, chromosome numbers. Friends, this clear-cut understanding is going to help us in solving various conceptual problems in this chapter. Invariably, any MCQ material or any question paper will have problems based on the concentration, the chromatid number, chromosome number. So I thought that this discussion will help us in having a clarity regarding these concepts before we proceed further, right? So with that, let's get into the discussion. Friends, as I said, today's concept is regarding chromosome number and chromosome concentration. So how do we have a clear-cut distinguishing thing regarding it, right? Say for example, we all know that a nucleus is supposed to have a DNA, right? This is a double-stranded helical structure, and you know that this DNA, since it has to get accommodated inside a nucleus, it is undergoing a sort of compaction which puts this double-stranded DNA into a structure called as chromosome, right? And your chromosome has got a primary constriction site which is called as a centromere. Okay, I hope you will be thorough with this basic thing. So this is I can call this as a chromatid, and this central part is called as centromere, right? Now what happens, friends, in synthetic phase of interphase, we have seen that the DNA replication happens. So from one double-stranded DNA structure, you get two such structure, right? You get two such structure. So in S phase, one double-stranded structure is going to duplicate into two double-stranded DNA. This is what is going to happen in synthetic phase of cell division, right? This we have seen this. So what happens is, so before that, see here in this picture you have, let's assume that the chromosome concentration, CONCENTRATION, chromosome concentration as C. Okay, chromosome concentration as C. Friends, how do you count the chromosome number? The idea here is you do it just by looking the number of centromere, which I have mentioned in a with a red color thing here. So the number of centromere is going to be 1. So number of chromosome is also going to be 1. So there is going to be 1 chromosome. Okay, 1 chromosome. Right? Now what happens is, there is a S phase, there is a DNA replication from, you can see here, one double-stranded helical structure to double-stranded helical structure. So what is happening here is a new DNA is formed, right? Thereby I could see there is in the same centromere there is an increase in number of chromatids. So here the chromosome concentration, since your initial concentration is going to be C here, the chromosome concentration now, that is post S phase, right? Post synthetic phase, the chromosome concentration is going to be 2C, right? And how do you determine the number of chromosome? By looking at by counting the number of centromere, right? Now you count the centromere, it is going to be still 1 here. I will mark it clear. There is going to be still 1 here. So the chromosome number remains same. So chromosome number remains same, that is one number of chromosome, right? One number of chromosome, but the concentration is getting doubled. And the chromatid number is also, if you consider this as A, this is going to be considered as B. So chromatid number is also increased. So mostly it is better, it is okay to have the idea of chromatid number also, but mostly we are questioned, okay, regarding the chromosome number and chromosome concentration, right? So this is how it is. Say for example, here is the situation. So again, what is the chromosome number and chromosome concentration in this picture, right? Friends, simple. Chromosome number, how many centromere are there? So centromere is going to be 1. So chromosome number is also 1, right? So 1 chromosome, right? So A, B, C, black one, pink one and green one. Hope the image is clearly visible. So initial chromatic concentration, chromosome concentration is C, which means that now I have C, C, C. So the chromosome concentration is going to be 3C. Okay, that is the chromosome concentration. Okay, chromosome concentration. Hope you get this point. And let's take one more situation. We have two different chromosome. See, chromosome number is going to be 0, 2 because you have got two different centromere. Okay, chromosome concentration is, initially it was C, now this is 2C, this is 2C. Okay, so each chromosome has got a concentration of 2C per chromosome. Okay, per chromosome, that is the chromosome concentration. So each chromosome has got 2Z. So 2Z concentration per chromosome is going to be the case here, right? So in summary, just count the number of centromere and tell how many chromosomes are there. Just count the number of chromatid, assuming that one chromatid is going to have a concentration of C. Count the number of chromatid and give the chromosome concentration accordingly. So that's the idea here, right? So when you have this understanding, any problem that you get in this chapter, provided if you have understood the further aspects of mitosis, meiosis clearly, any problem that you come across, okay, you will be able to solve it easily, right? So we conclude here, and we will now meet in another interesting discussion, probably with mitosis, the initial phases of mitosis. Okay, so I thought that before getting those things done, these concentration will really it really help us, to have a clear-cut understanding, right? Thanks for joining.